This audio lecture is going to cover information from Chapter 16. Chapter 16 has this elaborate title of Disruption of Healthy Tissue by the Adaptive Immune Response. Really what they're talking about is autoimmunity. So as we kind of touched upon in Unit 1 when we gave an overview of this whole entire semester, one of the disorders that we talked about of the immune system is autoimmunity. So autoimmunity is a failure of tolerance, and keep in mind that tolerance can be achieved in several different ways, right? So we can have tolerance that's achieved through in central tolerance, where we see the negative selection occurring to eliminate autoreactive T cells and B cells, lymphocytes. We can also have peripheral tolerance, right, where we have T regulatory cells that are regulating those immune cells that are out in the periphery and telling them don't respond. So when we have a disruption on those T regulatory cells, just like if we had a disruption in negative selection, then that could lead to a failure of tolerance. Now, when we look at autoimmunity, it can be organ specific or it could be systemic. So that helps to characterize and narrow down, along with other diagnostic tests, identify what type of autoimmunity you have, what disorder you have. It can involve antibodies, T cells, immune complexes, or combinations of these. So depending upon what is the kind of initiator, what is the cause, we can then label it as a different hypersensitivity. So again, autoimmunities, as I mentioned with the allergy lecture, autoimmunities do fall in as a hypersensitivity because it's an overreaction of the immune system, again, because there's some kind of failure of tolerance. Treatments vary, so it's, I'm going to say it's easier to treat. I don't know, easier is maybe not the right word. Someone who has like an organ-specific autoimmunity. When it's systemic, then you're trying to modulate the immune system to decrease that systemic effect. So in some cases, you're just giving back what is missing, what is affected by the organ. So we see that with like type 1 diabetics, they receive insulin because their beta cells have been destroyed. The beta cells are the ones that normally produce insulin in the pancreas. We can also look at using monoclonal antibody therapies. And again, if we have a specific antigen, specific antibody, we can use antibody against that specifically. One of the treatments that I've looked at to modulate the immune system is monoclonal antibodies that bind CD3. CD3 is a universal T cell marker, so the idea of this is it would affect how the T cells are becoming activated and then damaging tissue. There's also monoclonal antibodies that target cytokines, so TNF alpha, interferon beta. Those ones look at blocking the cytokines so that they don't have the effect of activating the immune system. So this is kind of a turned on the side partial panel of hypersensitivities. What has been removed is type 1 because that's allergies, but as I had just mentioned, there are autoimmune diseases that resemble type 2, type 3, and type 4. So again, keep that in mind, that again, we can have these different autoimmune conditions. You'll notice that type 2 diabetes is listed here, and there are subsets of individuals with what would present as type 2 that do have an autoimmune component to it. We would recognize that most type 2 diabetics, it's an impairment of the insulin receptor from being able to take up the insulin and function, quote unquote, normally. So it leads to it's a metabolic driven disorder. But again, some individuals might have an autoimmune component. Generally, when we're talking about autoimmune, with diabetes, we're talking about type 1, and you'll see that down on the type 4 category. Keep in mind, type 4 is where we have T cells. It's T cell driven. So we have T cells that recognize those beta cells that are found in the pancreas that produce insulin, so the beta cells get destroyed. I'm kind of partial to type 4 autoimmune disorders because I did research on type 1 diabetes. I also have special interest in multiple sclerosis, and we'll talk a bit about those. But again, keep in mind that there are other types. So like type 2, where we have antibodies binding against cell surfaces and antigens. So Graves' disease would be an example where the TSA, thyroid stimulating hormone receptor is targeted, and that leads to hyperthyroidism because the receptor's been targeted. An example of type 3 where we have immune complexes forming would be lupus. So in lupus, different factors can act as the autoantigen. So we can have DNA, RNA, and nucleic proteins that get targeted, immune antibodies binding that, forming complexes. So, and we'll talk a little bit more about lupus when we kind of switch gears and stop talking about the mechanism, but actually talk about different types of disorders at the end of the PowerPoint. So as I mentioned, tolerance is key in preventing immune responses against self. And so there's kind of two branches of it. So central tolerance, again, what's happening during the development of the lymphocytes before they have an opportunity to be activated. So in the bone, as you know, in the bone marrow and in the thymus, B cells and T cells are going to undergo negative selection. Really important to make sure that autoreactive T cells are deleted. Peripheral tolerance is what happens when they leave these primary lymphoid tissues and they have an opportunity to become activated. If they become activated normally and they are over-responding, this is where a cell could be told is induced to go in and become anergic and not respond. That would be part of peripheral tolerances, and again, T regulatory cells play a critical role in that. But if you have dysfunctional T regulatory cells or a lack of T regulatory cells, then there would be issues with peripheral tolerances mechanisms. So as we mentioned, negative selection of both B cells and T cells. Also when the thymus is expressing tissue-specific proteins during positive selection, in some ways that is also building self-tolerance. We have certain areas that are not part of the lymphatic system, that don't drain into the lymphatic system. So that actually helps with self-tolerance, so the brain, eye, and testes. And then also we see that with peripheral tolerance, we can induce anergic cells to become anergic if they're autoreactive B and T cells. And then again, the T regulatory cells are really important in autoimmunity. And so just to kind of expand upon that, as you guys know from our flip-flop paper, T regulatory cells, we would identify them as being CD4 positive normally, CD25 positive, FOXP3 positive, right? So those are some of the markers that we would use to identify them. And so those are naturally occurring in the thymus, but also in the periphery, and again, to help control autoreactivity. T regulatory cells inhibit on two different types of mechanism. They can have a dependent mechanism where they have to come in contact with the cell, and in that case, they provide a second signal that is inhibitory. So they use CTLA4. That's why they looked at that in the flip-flop paper to see whether that was being upregulated, because again, that's an inhibitory second signal that would then cause the other cell that the T regulatory cell is interacting with to become anergic or to calm down and not be so active. They can also have independent mechanisms where they don't have to be directly in direct contact. And so in this case, secreting cytokines like IL-10 is the major one that gets looked at, would actually cause cells in the nearby to shut down their responses. We also know that there's certain genetic factors that put you at risk of developing autoimmunity. Some of these are multifactorial, so it's not one gene that plays a role, but there are certain disorders that are monogenetic. So monogenetic is where there's a single gene causing that. And so APS1, autoimmune polyendocrine syndrome type, That is caused by a mutation to the AIR gene. And as you guys know, the AIR gene is important during that negative selection process. The AIR gene is what allows for cells to produce all of the different proteins that are going to be produced within the body and present those to the T cells and B cells. And so if you have mutations to AIR and this process doesn't happen, you're going to have failure of negative selection. So again, this is a gene that would directly impact negative selection. And if we impact negative selection, we're going to impact tolerance. So you can see these individuals that have this mutation, they have a higher frequency of different autoimmune conditions and also fungal infections because their immune system is being compromised. Another area that is researched looking at autoimmunity is HLAs. So HLAs are another term for MHCs. So again, these are the molecules that peptides get presented on. So certain HLAs increase your risk factor, so in susceptibility of developing autoimmunity. And we've seen this with a number of different autoimmune disorders like type 1 diabetes, but this list shows you some additional ones, and the allotype, so kind of like the serotype of HLA that puts them at risk factor. So these numbers correlate to a specific HLA, and again, you can think of this as MHC. And again, certain mutations on those, certain SNPs, single polymorphism changes, can lead to a higher risk factor of developing that autoimmunity. Also in autoimmunity, we see the incidences between male and female are not equal. So females tend to have higher rates of autoimmunity than males. And it's believed that the hormones affect the immune system. So in vitro studies have been done that look at hormones, show that hormones can affect the activation state of different immune cells. And so when we then move this into an in vivo type model, thinking of us as in vivo models, again, this is one of the exclamations on why women might actually have a higher rate of autoimmune diseases compared to males. Most studies are conducted only on female mice. When we look at autoimmunity, there are cases where studies are done on male mice, but again, usually all the mice in your study are either female or male. You try not to do a mix because again, you can't control for certain variables like hormones. There are a number of animal model studies, or animal models that are used to do studies. Most of these are mice or rats. So some of these models are spontaneous. So one of them that we use in type 1 diabetes is the non-obese diabetic mouse. So this has genetic factors that'll cause it to spontaneously develop diabetes at a high rate. So it's about 80% of the colony will develop diabetes by 6 weeks to 8 weeks of age. This mouse model is actually also used because we can take T cells from it and transfer it to other backgrounds of mice, and it can actually induce disease. So we can study these T cells. What antigens do they see? How does that fall in the epitope spreading, antigen spreading that occurs? So in a lot of these autoimmune conditions, it's not a single antigen that has been identified, it's multiple antigens. And so trying to find the sequence of what is recognized first and trying to prevent that recognition so you don't get recognition of later antigens that are targeted. Okay. And then there's also experimentally induced autoimmune diseases. So in some cases you can give a drug or expose them to the antigen, and that will then induce the autoimmunity. So you can use them. So you can kind of sync them. With the NOD mice, you can actually give them a drug that will synchronize. So if you were looking at an experiment where you wanted them all to have diabetes at 4 weeks, you can give them a drug, and I'm blanking on the name of the drug, but you can give them that drug, and they will actually synchronize and all have diabetes at the same time. There are a number of intrinsic factors and extrinsic factors that increase susceptibility to to autoimmune diseases. So there's roles for this different T helper cells in autoimmunity, and so autoreactive Th1 cells, they're known to secrete interferon gamma, and this has been shown to assist in the development of multiple sclerosis, but also other autoimmune diseases. When we look at Th17 cells, so that was one of the helper cells on the CD4 side that I was like, oh, you should, kind of be aware of, but that wasn't one of them that I was like, definitely know this, right? But they'll produce IL-17, hence their name, and they've been shown to play a role in rheumatoid arthritis and psoriasis. We also know that there are environmental factors that make it more favorable in developing autoimmunity. So we know certain diets can lead to a more likelihood of developing autoimmunity. It's believed that this causes a shift in the gut flora, and that the gut flora and the microbes present might actually play a role in causing the immune system become reactive, and then leading to autoimmunity. We also know certain geographic areas have higher incidences of autoimmunity. And so one of the theories is that vitamin D may play a role in how the immune system is responding and its activation, and then leading to diabetes. Obviously, more additional studies need to be done in order to kind of tweeze out some of this. And the role of microbes in gut flora is a relatively newer one, and understanding how microbes, your gut flora, affect your immune system is obviously is kind of newer area of research. In the end, induction is probably a multifactorial event, so not in most individuals with autoimmunity, it's not one single thing that led to it. So there'd be genetic factors that would lead to it, but then usually there's some kind of triggering event, and that triggering event is believed to probably have a number of different factors. So things like infection. With infections, microbes a lot of times use molecular mimicry, where they express proteins that are very similar to our proteins. So bacteria will have proteins that are very similar to ours. It actually allows them to evade the immune response. But if your immune system then targets that protein, because it's so homologous to your proteins, there can be this misidentification later on that then could lead to autoimmunity. Also, when we look at certain infections like viral infections, they're actually using your own cells to replicate. And so in that replication process and using your cells can get targeted because they're virally infected. That's a good thing. But because some of your own proteins can be presented then, you could have T cells and B cells recognizing that and then leading to it. We also know with some infections that can induce genetic changes. So in a virus taking over your cells and using them, there can be errors in replicating that genetic information that's yours with the viral genome. And so those genetic changes might cause changes to the proteins so that they start to look different to the immune system. It's not the same protein that was actually presented to them during negative selection, and so they start to go and target that. Also, when we look at things like damage, stress events, trauma, surgery, that can cause antigens to be available that had not been out in tissue fluid, and before had been contained in organelles within inside the cell, but now they're being leaked out. And we also know, food, if we change our diet and foods, that can alter the gut microbes. So again, that is an area of increase attention because we know that certain gut microbes can promote inflammation and hypersensitivity reactions, such as type 1 food allergies, but could those also end up leading to autoimmunity? So looking at infection, we can see here how we have this streptococcus infection, and so the bacteria is producing its proteins. We have recognition of those, so these antibodies are binding to the bacteria. That's great. It's going to clear the bacteria out, but these antibodies end up cross-reacting to proteins that are found in heart tissue, and so now you have immune system recognizing that. And so this is why for some individuals after a strep infection, they can then have heart damage and recognition, which we'd recognize as rheumatic fever. We also know, as I mentioned with trauma, there's cases where, if you had trauma to an area of the body, this example here is in the eye, because the eye is damaged, there's going to be proteins that are going to leak out. Those go into the draining lymph nodes, or they're picked up by dendritic cells at the site of the trauma. And then they go back to the lymph nodes, and those are presented to T cells. T cells recognize them because, again, they haven't seen them. Maybe there's been some kind of change to them. So it looks slightly different than what they were presented to during negative selection. And now you have T cells that are recognizing those proteins that are expressed in that damaged tissue. And that could lead to damage. And so again, in here, individual might have lost sight in one eye because of some kind of traumatic event, but now they lose sight in both eyes because the immune system comes in and responds to that tissue as foreign, even though it is self. So there's a number of infections that have been associated with autoimmunity, and so I want to remind you that for autoimmunity, you have to have recognition of self. So just because you had this infection doesn't mean you have autoimmunity. It doesn't mean you will have autoimmunity. Again, we recognize that there are going to be genetic risk factors that would you need to have those genetic factors that would put you at risk of developing autoimmunity. And so if you have these infections and you have those risk factors, then you may end up developing autoimmunity. But again, you need to have recognition of self. So these individuals would have to have antibodies that would recognize self to have autoimmunity. So again, having the infection does not mean you have autoimmunity. I emphasize this because you'll see Borrelia on here. Borrelia is the causative agent for Lyme's disease. And so again, you see that individuals that have certain HLAs put them at risk of potentially developing autoimmunity to it. Again, just because you have Lyme disease does not mean you're going to develop autoimmunity. Some patients, and right now a small number of patients, have been shown to develop anti-nuclear antibodies, which are abbreviated ANA. So if you had Lyme's disease, if you're starting to have symptoms that would indicate that you're having an autoimmune condition, they may run an ANA. It's a pretty general antibody that they're looking at, and it's an indicator of not a single autoimmune condition, but there's a number of autoimmune conditions where we see these anti-nuclear antibodies being formed. So again, not everybody that has Lyme's has this. It would be someone who's then presenting with autoimmune type conditions. Okay, so we want to keep in mind when we have an infection, especially a chronic infection, we're going to have chronic inflammation and, kind of question, does this cause that autoimmunity to go from an infection to autoimmunity? Is it molecular mimicry? Because in using and being in the host, it's maybe using some of the machinery, especially for like a viral infection like Coxsackie virus. And so that might lead to molecular mimicry, or does it lead to an epitope spreading or cross-reactivity with the antibodies that are being produced? So it's kind of like a chicken and egg problem. Is the infection just the trigger that causes it because they have genetic factors, or is it not? Is the autoimmunity caused by the infection, right? So it's kind of a nuance, a play of words, right? But again, kind of a chicken and egg problem. Is the infection the chicken? Is the autoimmunity the egg? And which came first is something that needs to be looked at and tweezed out, and looking at the mechanism of this, right? We also know that the binding of the antibody to cell surface receptors can cause autoimmune disorders. So there's a number of autoimmune disorders. And again, keep in mind that I do want to point out type 2 diabetes is on here. Not all individuals with type 2 diabetes have autoimmunity, just like not everybody with Lyme's disease has autoimmunity. Some individuals that have type 2 diabetes can develop autoantibodies to the insulin receptor, right? So they would have an autoimmune component. A classic example of where we have antibodies to a surface receptor is the thyroid stimulating hormone receptor with Graves' disease, which I already pointed out. I'm including these tables that are from the other immunology textbook. It just goes through and identifies some of the different diseases. I like these two slides that I'm going to show, this table, because it breaks it down to organ-specific autoimmune conditions. So you can see some examples of that, where there's a single tissue organ being targeted, and even in most cases, a single cell type that's being targeted. So like type 1 diabetes, it's not all the pancreas that's been targeted, it's only the beta cells that are being targeted. And then we have systemic, where the effect is more widespread. Sometimes that's because the central nervous system is being affected, and so because the central nervous system affects all of our other, organs and how they function, it can have a systemic effect. In other cases, because the antigen is found systemically throughout the body and the immune system can interact with it, then we can see a systemic effect. All right, so now we're going to look at some of the different autoimmune conditions. So the first one we're going to look at is Hashimoto thyroiditis. So this is an autoimmune condition where autoantibodies and Th1 cells recognize thyroid antigens, more commonly in women. Antibodies interferes with iodine uptake. Iodine is important for the function of the thyroid. It's one of the reasons why iodine is added to salt, so that we consume enough iodine so our thyroid works functionally. This leads to decreased thyroid function and hypothyroidism. Hyperthyroidism causes a slowing of metabolism, so individuals sometimes will experience weight gain. This can induce a DTH response in the thyroid and lead to inflammation, which results in goiters. So you can actually get this enlargement of the thyroid gland. So here we're looking at the lymphatic tissue. So there's an increased number of cells that are present there. The next one that we'll look at is lupus. So with lupus, or SLE, this is where immune complexes will be created, and then they end up being deposited in the glomerulus of the kidney, and this causes kidney disorders. Again, more commonly in women, so it's a 9-to-1 ratio in women, and typical onset is between 20 to 40 years of age. It's more frequent in African American and Hispanic women than Caucasians. The autoantibodies that are produced bind to DNA, histones, and other cell structures. And again, what happens is they create immune complexes. Your body tries to remove those immune complexes. The way that does that is by increased urine output. So this gets the complexes end up concentrating in the kidney, in the glomerulus, because they actually can't pass through the glomerulus, and that then causes kidney damage. Symptoms symptoms that happen are fever, weakness, arthritis. You can get skin rashes. Lupus is known for having this classic butterfly skin reaction on the face. And part of that is like an immune system reaction that's happening. And again, kidney destruction would be dysfunction would be one of the more severe negative consequences of lupus. You can see here with this figure from the textbook, it looks at how you can get epitope spreading. And so here on the left panel, you have your CD4 T cell recognizing its antigen. It's going to become activated, and it's recognizing this H1 peptide. But you can see how there's this kind of complex that's formed. So that's going to then activate a B cell, which we're seeing on the right side. Well, now this B cell is going to be interacting with all of these other T cells. So we have that original the H1, but we also now have interactions with H2, H3, and we could get additional interaction with other components in this complex, so that these T cells would become activated. They would then activate B cells and then produce antibodies to them. So this process where we go from recognizing like a single epitope to multiple epitopes, which is commonly seen in autoimmunity, is known as epitope spreading. Rheumatoid arthritis, or RA, is another autoimmune condition. So this usually has a diagnosis onset of 40 to 60 years old, more frequently in females. And what happens is we have this chronic joint inflammation. There's rheumatoid factors that are produced, and then autoantibodies react to those. They form immune complexes. Inflammation is going to be happening in the joints, and you can see in this individual how the joints start to become disfigured and not as functional. And so usually treatment is with anti-inflammatory drugs and steroids to help reduce that inflammation. There has been development of some more specific anti-cytokine antibodies that have been proposed to be introduced. Just want to point out that there are a number of autoimmune conditions that are rheumatic diseases and that can induce arthritis. There is recognition that rheumatoid arthritis is kind of a catch-all, and many people with other autoimmune conditions get diagnosed and identified as having rheumatoid arthritis, but the root cause of their rheumatoid arthritis might be other autoimmune conditions. And those are shown on that list. Then type 1 diabetes, as mentioned in some of those earlier panels, type 1 diabetes is where we have the insulin-producing beta cells being destroyed. So here we can see this islet. So you can see these cells have more of a cloud, puffy appearance to it compared to the nearby tissue. And so the islet has beta cells in it that produce insulin, and those end up getting targeted by CD4 T cells and antibodies, and they get destroyed. Type 1 diabetes is also called insulin-dependent diabetes because they require insulin in order to survive. So because the beta cells have been destroyed. It's also referred to as juvenile diabetes because onset is usually young, so under the age of 14. Now, onset can occur at older ages and can occur after traumatic events. So there are cases, in not huge numbers, but there are cases where individuals have undergone surgical procedures and then afterwards end up developing type 1 diabetes, believing that that surgical procedure caused trauma, caused the release of antigens that then led to the autoimmune condition. So as I mentioned, generally under the age of 14 is where we see most cases of type 1 diabetes, and we can see cytotoxic lymphocytes, CTL. that infiltrate into the pancreas, and we also have activated macrophages. So if we did staining and looking at markers for these different cell types, we would see them present in the beta in the islets where the beta cells are found in the pancreas. There are, for diagnostic tests, besides blood glucose levels being elevated, responding to insulin to bringing that back down. Being of young age, additional diagnostic tests would look at autoantibodies that are present. And so we have kind of some non-specific general ones like islet cell, cytoplasmic autoantibodies. So this would be like a lysate of the cytoplasmic fraction of islet cells. So there's going to be a lot of different components in it. We also usually, individuals will be positive for insulin autoantibodies. It's not necessarily believed that this is the first antibody that's formed in it. They can have other autoantibodies that would be formed before the recognition of insulin. I did add a slide on multiple sclerosis. So multiple sclerosis occurs in women 2 to 3 times more frequently than men. But I do want to point out that men do still experience autoimmune conditions. And obviously the mechanism in which they develop it, if hormones are playing a critical role, we also need to look at them. Do they have risk factors, genetic risk factors that are putting them more at risk? So even though they may not have hormones that are increasing that risk, do they have other components that are increasing the risk? All right, back to multiple sclerosis. So this causes neurological disability, and so it's the most common cause of neurological disability associated in Western countries. It's more common in the Northern Hemisphere, and again, some of this is believed that vitamin D might play a role in this for it. We also know that certain infections may put individuals at predisposition of developing MS, multiple sclerosis. Up in this figure, you can see how a normal nerve is going to have myelin sheaths that are covering that axon. And then what happens is the immune system ends up destroying it, so it kind of short circuits the information. Those action potentials aren't relayed down to the terminal ends of the axon. And so when that information doesn't get relayed, again, your ability to have control of body movements, use of your limbs, and how your brain is also processing that information as it's returning to the brain is affected. So we see autoreactive T cells that form lesions on these myelin sheath in the brain, but also in the spinal cord. So individuals with MS would routinely, have yearly MRIs to determine whether there's an increase of lesions or just a kind of status quo of the number of lesions they have. And depending upon which neurons are affected, that can relate to the different symptoms, also the severity of the symptoms. So it can range anything from a numbness to actual paralysis or loss of vision. If you go to the National MS Society page, which I provide the link because that's what the image is from, they have a symptoms page and a list of different symptoms. And again, pretty much anything that your central nervous system controls, it can be impaired by multiple sclerosis. This doesn't mean that people can't lead a good life with multiple sclerosis, right? So there are a number of different treatment strategies, drugs that look to decrease the formation of new lesions so that additional functions wouldn't be lost. Our last autoimmune condition that we're going to look at is celiac disease. And I'm just going to start off with, I disagree with this statement from the textbook, right? So the textbook has a statement of celiac disease is an hypersensitivity of food that has much more in common with autoimmune disease. Wrong. Celiac disease arises from having a gluten allergy, which is a type 1 response, and that leads to autoimmunity. Celiac disease is an autoimmune disease. It is not, it is more common, it has much in common with autoimmune disease. That means it makes it sound like it is not an autoimmune disease. It is an autoimmune disease. Just because you have a gluten allergy does not mean you have celiac disease. Gluten allergy itself is a type 1 allergy. Celiac disease is that gluten allergy has led to the immune system recognizing self. So this is similar to like the Lyme disease, the infections, right? So think of like the gluten allergy as the chicken or the egg, right? And then celiac disease now is the downstream effect of that. Okay, again, individuals would have risk factors that put them at risk of developing autoimmune conditions, celiac disease, but that gluten allergy, because the immune system is targeting the gluten, it ends up accidentally targeting self and the tissue in the digestive system. So you can see in the figure how we have our normal intestinal cells, nice villi here with these nice columnar epithelial cells present. And then we can see in the celiac individual how those structures are not present. Keep in mind that on the normal side, these epithelial cells are important for absorption of this nutrients, right? And so on the right side, we're not going to have effective absorption of nutrients, right? Because we don't have these nice columnar cells, columnar epithelial cells to absorb the nutrients that's in the GI tract. So one of the side effects of celiac disease is different vitamin deficiencies because you're not getting effective uptake of nutrients, right? Along with the inflammation that's occurring, along with the allergic reaction to gluten, and then also the autoimmunity that is to self tissues. So you can see some of the different antibodies like TG autoantibodies that are produced in celiac disease. So again, these are your own tissue, your own proteins that are being recognized. So again, celiac disease is an autoimmune condition. It is not like an autoimmune condition. It is not mimicking an autoimmune condition. There is recognition of self. And again, just because you have a gluten allergy does not mean you have celiac disease. You would have to do the diagnostic testing to see that you actually are producing autoantibodies to this. Just because you have a severe response to gluten does not mean you have celiac disease. It's the production of these autoantibodies that then makes it autoimmune so that you have celiac disease. So again, when we kind of look at the mechanism, there's going to be recognition of gluten as being an allergen, where we're going to get a type 1 response. So we're going to have IgE antibodies that recognize the gluten. Because tissue transglutamase, glutaminase binds to the gluten, it's now there when the immune system goes to target the gluten. And so again, if you have certain risk factors like HLAs that put you at risk of having autoreactive cells, you could have an autoreactive T cell that then recognizes that tissue transglutamase and then develops antibodies to it and then starts to destroy the tissue. This figure could use a little bit better feedback to the scientific illustrators because somehow villi all of a sudden form. It's not the best figure, but again, the idea, keep in mind, it's a sequence of events that lead to the autoimmunity. And again, the triggering event for individuals with celiac disease would be that gluten allergy. If you could avoid them developing the gluten allergy, they most likely would not develop celiac disease. They might develop some other autoimmune condition because a lot of the HLAs are common between HLAs that put you at risk factors are common between different autoimmune conditions. It doesn't mean HLA doesn't mean this is the autoimmune condition you're going to get. In most cases, it makes it more likely for you to develop an autoimmune condition. And it's the triggering event that kind of determines what is the autoimmune condition that the immune system is going to, what are the autoantigens the immune system is going to target, thus the autoimmune condition. If you do have any, hopefully this gives you an overview on autoimmunity. Autoimmunity could be a whole entire semester. So just like allergy could be a whole entire semester or a whole entire career, right? So if you do have any questions, please do not hesitate to contact me, ask questions. We will be covering some of these key slides, and you'll have an opportunity to ask questions on Tuesday in class. But remember, this is the end of the semester, so quick, short unit with the time that we have left. So have a great day.